And it looks like we have our attendees joining us. I'll give it about 20 seconds so that everyone that was in the waiting room can join. Still see our numbers coming up a little bit, so I will wait just a few more seconds.
Right.
All right. I want to welcome everyone to our Mesulam Center Alzheimer's Disease Seminar Series. Just a few brief housekeeping rules. We will have the first presentation by Dr. Gratton, and at the end of her presentation, she will take questions. We'll have about 5 or so minutes for questions for her. And then at 12:30, we'll have Dr. Pancani give his presentations, and any questions for him will be taken at the end of his presentation. And if we have any additional time, we can open it up to the floor for questions for either. And so with that, I will, and then also a quick reminder, you can put your questions in the Q&A box, or if you'd like to ask questions in person or speak your questions, you could raise your hand and I can move you into the webinar. And with that, I will turn it over to Dr. Disterhoff.
Good morning. Welcome, everyone. I'm John Disterhoff in the Department of Physiology. I chair the ADC pilot grant program, and each year we ask the recipients of the pilot of our ADC pilot grants, and we have 2 of them each year, to give an update of the progress they've made on their research topic. And the recipients, obviously not of this year, not the 2021 recipients, but the 2019-2020 recipients. So they've had an opportunity to actually make some progress on their project, although I'm sure we're going to hear from Dr. Caterina Gratton that there's been some limitations because of COVID. But in any case, and this is a reminder also to all of the community that we will be having, we plan to give 2 more pilot awards next year. So look out for the announcement, the solicitation for those awards. So our first presenter today, just very briefly, is Dr. Caterina Gratton. She's an assistant professor in the Department of Psychology. She has a very stellar preparation before she came to Northwestern. She worked with Kara Federmeyer at the University of Illinois, Urbana-Champaign as an undergraduate. And she worked with Mark Desposito and Michael Silver at UC Berkeley, where she did her PhD. And then she was a postdoc for 4 years with Steve Peterson at the University of Washington in St. Louis. She currently has an R01 recently awarded from the NIMH, and her pilot project, which was entitled Precision Scanning of Functional Brain Networks in Older Adults, will be the topic of her presentation today. I think this, we'll see that this has potential relevance for understanding, for example, brain networks in aging individuals who are, for example, superagers as compared to normal agers. That was a particularly interesting thing for the committee, and I'm sure we're going to hear about that as well. So Dr. Gratton, would you like to begin?
Thank you for the very nice introduction, and that's a perfect lead in. Let me just share my screen. And let's see, can you see my main slides right now?
Yep.
Okay, perfect. So I'm very excited to have the opportunity to speak with some of you today about this pilot project that was very nicely funded by the Mesulam Center, looking at precision fMRI and its ability to map and visualize brain organization in older adults. I thought I would start off the talk by just giving you a little bit of a broader overview of the types of questions we're interested in the lab. So we also, both you know where I'm coming from in terms of asking these questions, and maybe we'd have the opportunity to connect at other moments in time. And the lab is really focused on this, I think, really remarkable human ability to engage in different thoughts and actions depending on our goals. And this is true not just in normal everyday world, but even in the relatively constrained environment many of us find ourselves in right now with the pandemic. Even so, sitting at our desk, we could choose to engage in many different tasks. We could choose to, for example, watch the very salient Zoom presentation in front of you. But you might also choose to think about a meeting you had yesterday and write down notes regarding that, complete the motor actions to drink a glass of water, read a really interesting article you recently found, or maybe even, if you're like me, obsessively smell things around the house in an effort to determine whether or not you've developed anosmia. And so this ability to engage in different thoughts and actions, depending on our goals, is broadly under the umbrella of what I will call control. It's very related to concepts of executive function or cognitive control. And I'm particularly interested in the neurobiology of control, which I would argue is quite intriguing from a mechanistic perspective, because it requires both that some brain regions maintain and represent current task and goal parameters, as well as coordinating functions that we know are encoded in many other regions of the brain, which fundamentally then requires an understanding of large-scale brain networks and how they're organized. And so the lab has different lines of research that proceed along these various lines, both in terms of understanding control systems themselves, understanding these large-scale brain networks and how they change, and also understanding these not only from the lens of looking at young, healthy adults, but how they vary and are disrupted, and in particular, how they vary over the course of the lifespan, as control functions are known to be among the last to develop and among the earliest to show declines in aging. And so today I'm going to give you a kind of very brief snapshot, both into the methods that we use and then into these new precision fMRI techniques that we've been trying to pilot recently in older adults. And so for this talk, the primary tool that we'll use is functional magnetic resonance imaging, or fMRI, to measure brain activity with the bold signal, which tends to increase with increases in neural activity. Now, the main advantage of using fMRI for these types of questions is that it gives us a picture of brain activity all over the brain approximately simultaneously, which makes it really well suited for measuring large-scale brain networks. Now, the way that we measure these cross-system interactions is just by taking a measure of statistical association between different brain regions, often simply by just correlating their activity patterns. So for example, if we take the brain activity in left and right motor cortex over time, you might see that they'd have a very similar activity pattern, whereas another region that's part of a different functional system, like left visual cortex, would have a very distinct pattern. Now, what was surprising when this was first observed is that this is not only true when people are completing a movement or some other motor task, but also when people are asked to sit very still at rest, just staring at a fixation cross. Yet you see correlations in the spontaneous activity of these different brain regions. And in fact, these spontaneous correlations, which correlate across a broad motor system of cortical and subcortical regions, mimic the patterns of activations that you see in different motor tasks. Now, this is true not just for motor-related regions of the brain, but many regions throughout cortex and subcortex. And I can represent all of these patterns together at once in the form of what's called a correlation matrix, where any cell in this correlation matrix just represents the correlation between a pair of brain regions. Now, here I've arranged the regions randomly, so they look a little bit like snow. But if I reorder them such that I put regions that are part of the same system close to one another, you can hopefully see this really beautiful structure emerge. It's often called the block diagonal structure, where you can see that there are high correlations along the diagonal that is within a given system, and low correlations in the off diagonals, that is between different systems. Because of this, we can then very effectively use many different community detection algorithms to identify systems of the human brain. And for example, I can mark them off with red lines here, or I can paint them on the brain in different colors as shown here. And so what you can see here is that this approach, which is data-driven and done non-invasively in a healthy human, can be used to identify systems of the brain that both recapitulate previous things that were known about the brain, like the presence of the visual and the motor-related systems, as well as being able to identify higher-level systems, including those involved in control, like the frontal parietal system, as well as the better-known default mode system, or the well-known default mode system. So this is very exciting. A lot of the work that's been done thus far has focused on making these measurements in large groups of individuals. So these plots are actually 120 individuals averaged together. But recently, there's been interest in trying to extend these to understand other populations. So for example, one question might be is whether this systems level pattern stays preserved across the lifespan or if it changes with age. And in particular here in my lab, we're interested in trying to understand whether or not changes in the systems level organization might be related to changes in cognitive performance that are often seen in older adulthood, and that are seen particularly for fluid functions like those control related functions I mentioned earlier. And so some past work has been done to examine this question. One common finding from this past work that my collaborators and I have also replicated shows that one of the things that you see with aging is a decline in the segregation of these systems. So if you compare these correlation matrices in, say, young adults from ages 20 to 34 and to progressively older groups until older adults from 65 to 89, what you might notice is that there tends to be a decline in that block structure along the diagonal, less correlation within the networks, and an increase in the correlations between different networks. And so this leads to an overall decrease in the segregation of systems with age, which may suggest that there is a decline in the ability of these systems to functionally segregate from one another with time. And you may note in particular that this is stronger for some systems than others. It's particularly been noted to be strong for control-related systems of the brain. Now, one thing you might observe in looking at this graph is that although there is this general trend line, there are also substantial individual differences in this decline, with some individuals showing very preserved segregation and others individuals showing much more prominent decline even in earlier time windows. And so this is true not just for these brain system measures, but has similar things have also been observed for cognitive measures, including measures of executive function or working memory as shown here, that while there's an overall pattern of decline, some individuals actually show relatively preserved working memory functions, for example, and others decline much more rapidly. And so here we wanted to understand whether these two factors might be linked, but to do so really requires producing a very reliable measure of a single individual's brain networks. And so this is to determine whether these differences are real or just a function of noise in our signal, we need a way to reliably map these networks at the level of individual subjects. And this is a topic my colleagues and I have recently been working a fair amount on in developing a new technique that we call precision fMRI. And so I'll tell you about that in the next couple slides. So as I mentioned, many typical studies in the field of resting state brain networks take small amounts of data from each individual, something like 5 to 10 minutes, and pull them together to create a group average picture of the brain. And this helps to overcome high noise and low signal that we might get from typical functional MRI methods. And with this approach, you can show that you can produce very robust pictures of brain networks across these groups. For example, these are two different studies collected at WashU and at Harvard, which produce very similar pictures of what the typical brain networks are on average. However, it doesn't, of course, give us a picture of what each individual subject looks like. And so to address this, my colleagues and I have been developing a novel technique that we call precision fMRI. It's also been called deep or dense scanning. You may have heard it under those terms. And the basic premise of this is to flip the original sort of paradigm on its head, and rather than collecting a small amount of data on many subjects, you collect a large amount of data on a single subject, ideally across multiple days and different task states, coupled with many other phenotypic variables and sort of extensive denoising techniques to really try to get as good of a picture of that individual subject's brain. And so this has been spearheaded in a couple of other studies. The original study that inspired us to do this work was conducted by Russ Poldrack, who is now at Stanford, at the time was at UT Austin. And he collected his own brain networks twice a week for an entire year. And so this produced a very large amount of data. In addition, had blood draws and other measures taken in order to sort of produce a very robust and detailed image of his own individual brain. This is called the MyConnectome project. Shortly after that, at WashU, we collected another data set that was somewhat similar. It was instead of just one person, it was across 10 different people. And each of these 10 participants completed 10 separate functional MRI sessions, which is called the Midnight Scan Club data set. And with this very extensive amount of data per subject, many hours of data per subject, we can now focus on other aspects of the data that will help us hopefully achieve better clinical validation and move these measures into the scanner and understand individual subjects in a better way. And so this includes having a better sense of the reliability of our different measures, their stability across days and across different types of task states, and also individual differences and their sort of meaningfulness and their characteristics across individuals. And so just to give you a little bit of a snapshot of the types of information that we've obtained from these measures, here I'm showing you test-retest reliability of these functional network measurements in the MyConnectome dataset from Russ Poldrack. And what you can see here is that with functional connectivity measures, you can achieve actually very high reliability, above 0.9 reliability in test-retest, but it needs more data, substantially more data than what has typically been collected. So you need at least 30-ish minutes of high quality data from each participant in order to achieve these high quality measurements. This was done in just one subject, but we have now replicated this pattern here shown in the Midnight Scan Club data set, and several other data sets are starting to replicate similar results. We've also, in one of the projects I did on this data set, we've also looked at their stability across different days as well as different task states, asking whether or not functional networks change in meaningful ways depending on what you're doing or what you're thinking about or what day it is. And so we used that Midnight Scan Club data set, which had data from 10 different days, as well as from 5 different types of task states to address this question. And by comparing the functional networks, those correlation matrices from each subject, each day, and each task state. And I'll just sort of jump to the chase because I want to tell you a little bit about the older adult data, but to just say that what we ended up finding, and hopefully you can see it at a glance, is that these network patterns are really dominated by stable features, both commonalities that are seen across the group. You'll note the background is around 0.5 or 0.6, as well as individual specific features. That is, a particular person's brain network measurements look most like themselves, regardless of what day it is or what task they're completing. We've also used this approach to then hone in on those individual features and try to understand them better. And so what we've done, for example, is compare the network patterns for a particular location in one subject to that same location in a large group of other individuals. And what you find is that if you take the spatial correlation between the two, you'll find locations where it looks very different. In these two, you might note, for example, the negative correlations in lateral frontal cortex. Resulting in in locations that show very distinct patterns from the group average. And so we've now been able to do this across many groups of young, healthy individuals, and been able to show that each person shows these individual variants in their network organization. So I think this work is exciting in terms of its potential clinical applications, both for understanding differences between normal and pathological pathological brain in the face of heterogeneity that we see in many different neurological and psychiatric disorders, and also for creating tools then for accurate diagnosis and prognosis. And already several studies have shown that these more individualized measures outperform typical group measures in predicting the course of different disorders. And finally, or thirdly, we can also use these to guide individualized interventions. So there's been several papers now showing both with transcranial magnetic stimulation as well as deep brain stimulation that using individualized measures of functional connectivity can predict locations that are more likely to have consistent results across subjects or inconsistent patterns. And finally, we can use these measures to track the efficacy of treatment and try to understand what treatment is exactly doing to underlying brain organization. It's a recent, very neat study that was done at WashU looking at casting the dominant arm and seeing what effects there are on functional networks after this sort of approach. So in this last part of the talk, I want to tell you about this recent pilot project that we've been doing, trying to extend these measures into older adulthood. So everything I've shown you so far has been done on relatively young adults. I think the oldest were in around 40 that have been participating in these precision fMRI scans. And so here we wanted to ask whether it is possible to collect this sort of data in older adults and whether we would achieve similar measures or if there are any distinct patterns in this age group. And so in the pilot, our goal is to collect 10 subjects between the ages of 65 and 75 and have each of those 10 participants complete 5 separate fMRI sessions focused primarily on resting state fMRI data as well as some structural measures. And they also completed some behavioral measures outside of the scanner, both in the lab as well as in take-home questionnaires. Now, because of COVID-19, we were interrupted in our data collection, but we were able to collect data from 6 participants who completed somewhere between 1 and 5 MRI sessions each, which already allows us to address some of the questions that we were interested in looking at. First off, it allows us to address the feasibility of collecting these high quality measures while carefully addressing common artifacts that you see in fMRI related to motion and respiration and other physiological effects. And what we found is that quite pleasantly, we were pleased that we were able to collect very high quality data from these participants. We didn't lose that much data due to motion. And we were able to achieve over 30 minutes of high quality data from each session with reaching up to about 150 minutes for completed participants. And we also, along the way, sort of addressed various aspects of motion and respiration that are specific to older adults. And I'd be happy to talk with anyone offline about that who might be interested. Because of this large amount of high quality data, we are then able to make very high quality images of individual subjects' brain network patterns. So this is one of our participants, a 67-year-old female. And you'll note that they have a very nice block diagonal structure here, much like I talked about earlier. That earlier map was on the group average. So seeing something like this at an individual subject level is usually an indication of very high quality data. And in fact, is very similar to what we then see in our young adult participants in the lab as well. We can also use this data to look at the reliability of these network measures in older adults. And we can show that, again, here we can achieve very high reliability in our network measurements, above 0.8 with about 30 minutes of data and above 0.9 with 85 minutes of data. And this is similar to, again, what we've seen in our young adult data sets. Finally, we've been looking at day stability in these functional network measurements, comparing these functional networks across the 5 different or less days that participants completed. And what we find here is, again, consistent with what we saw with our young adults, where the participants look very much like themselves, regardless of what day they come in, and much less like other subjects, indicating that they also have these large individual differences that distinguish one subject from another subject. So just to preview where we're going next with this, our goal now is to identify these reliable individual differences in brain organization in older adults, much like we've seen with young adults, and compare their patterns to see if they occur in similar locations and have similar features as we've seen with young adults. We also are interested in tracking longitudinal stability beyond just the 5 sessions that were completed here, but in the future over the course of multiple years. We've looked at this a little bit in that MyConnectome data set from Russ Poldrak, where we see very high longitudinal stability in in younger ages between the first 3 weeks and last 3 weeks of the year. And here we're interested in seeing if we see something similar in this period of time where there can be more behavioral changes. And just as a preview, we've already started looking at this in some of our young adult data sets that we've collected here at Northwestern, where we've collected one participant pre-pandemic and then another mid-pandemic at some point more recently. And we've been able to show that, excuse me, much like we saw with the Brust-Poldrack data set, we see a high degree of stability, even with a pandemic intervening, strong social changes in these brain network organizations, and that distinguish this individual from another individual that we've collected. And finally, we're interested in asking whether or not these any changes that we observe are related to behavioral changes that are seen in these individual participants, and if they help us to track participants that are likely to stay stable in their behavioral and cognitive phenotypes, or those that may show greater declines. And this is something that in the past we've been able to show in our young adult data sets, that these individual features can be related to differences in behavioral measures that are collected outside of the scanner. And so, as I said here in particular, our goal will be to look at whether or not it's related to control related functions and how they change. So that's the end of what I had for you today. I want to close by thanking our funding from the Mesulam Center and also my lab for doing this work. And I particularly want to highlight my student, Deanna Perez, who's circled here, who's done all of the data collection and analysis for this project. And I put both of our contact information up here in case you want to get in touch with either of us about these projects.
Thank you very much, Caterina. I think we'll take about 5 minutes for questions. I'm not, Lisa, how do we see the questions here?
I don't see any questions right now. So if anyone has any, feel free to put them in the Q&A box, or if you want to raise your hand and I can let you in, we can also have you ask your question.
Well, let me ask one question. So, Caterina, you mentioned during the talk that there are certain control systems that are maintained and other systems that tend not to be maintained. And can you give us some examples of that, just to what you mean by that?
Sure, with age, you mean?
Yes.
So let me go back to those systems. So the general pattern is that control systems in general tend to show more of that desegregation. Maybe I'll get there eventually. So I don't have the colors. I do have the colors labeled. That's good. So like this yellow system is the frontal parietal system, and this teal system is the ventral attention system. So those are two where we see particularly prominent changes with age. The default mode system has also been implicated in very prominent changes, though often more associated with Alzheimer's disease pathology. So a lot of higher level association related systems show this segregation more than, say, lower level systems related to motor or visual function. And that's been a fairly consistent pattern, although of course it'll differ with pathology. I have another set of projects that I've done in collaboration with Joel Perlmutter and Megan Campbell at WashU, looking at individuals with Parkinson's disease. And there what you actually do see is a lot of disruption in motor related networks of the brain, perhaps not surprisingly, but you don't see that as much in typical aging.
So in those people, the control system is disrupted or not
both
maintained.
Control and motor systems in that case. Okay. Yeah. So it's a little bit, it's found in more different locations, whereas here it's more this desegregation of just the control systems.
Okay. Lisa, any other questions?
Sure. It looks like we do have one question from Dr. Braga, and his question is, what kind of network properties do you expect to be altered or preserved in superagers?
Yeah, I think that's a great question. I think based on some of Emily Rogalski's work, I would be really interested in looking at those control systems in particular, some of the locations that they've seen structural differences between the superagers and non-superagers are in the middle of what we think about as part of the cingulopercular system. And so it would be particularly interesting if there we're seeing really preserved, say, cingulopercular network structure relative to other networks. I think that would be like a fascinating thing to follow up on.
All right, and then also I did have Dr. Mesulam that wanted to ask a question, so I've added you to our panelists. You can go ahead and ask your question.
So Caterina, beautiful, beautiful work. very interesting lecture. I had a question for you. You know, it's interesting. Different words mean different things depending on the context. You know, in some, you use the term segregation. So in some context, segregation is not good. So what is the actual evidence that in the course of physiological aging, not in disease system, in physiological aging, that loss of segregation is correlated with decline of function?
So this paper, for example, looked at this question in a relatively large data set. I can't remember exactly how many people they had, 150 or so. And they showed that this change in segregation was related to declines. I think it was particularly in memory-related functions in this case, on the group average. So I think there is some evidence that at this level, desegregation may not be good. And in young adults, there's also evidence that good segregation is often related to better behavioral function in tasks, although what you see in tasks is then a departure from that, right? That the tasks start with this very segregated modular structure, and then the tasks cause more interactions to happen. So there may need to be a little bit of flexibility in that organization as well.
Okay. All right. I don't think I see any other questions in the chat or the Q&A.
Great. So why don't we go on with our second presentation? Can people hear me? Oh, there we go. So our second presenter, and I should mention that each year for our pilot projects, we attempt to give one pilot project for a kind of a human-oriented type of research, and that would be Dr. Gratton's. And the second one is more basic, what we call basic science broadly. And so, and you'll see that's Dr. Pancani was that awardee in 2019. So Dr. Tristano Pancani is going to talk to us today on basal forebrain cholinergic neurons in aging and proteinopathies. So Dr. Pancani his initial training was at the University of Florence in Italy. Then he came to the United States, initially at the University of Kentucky, Department of Biomedical Pharmacology, where he worked with Olivier Thibault and Phil Landfield, both for a PhD, and then stayed for a couple of years as a postdoc. He moved from there to Vanderbilt, where he was a postdoctoral fellow with Dr. Jeffrey Kahn, and then came to Northwestern initially as a postdoc in the lab of Dr. Jim Surmeyer in physiology, Department of Physiology, and Dr. Pancani is now a research assistant professor in the Department of Physiology. He does in vitro work, uses a lot of calcium imaging as well as biophysical recording. So Tristano, take it away, please.
Thank you so much for this generous introduction, actually. Let me see if I can figure out how to do this. Can you see me?
Yes.
Awesome, perfect.
You need to put it on. Yeah, there we go.
Yeah, it's going. Yeah. So actually, first of all, I want to thank you guys, all of you, for this opportunity. It actually, it gave me the opportunity to go back to my studies I did in actually grad school, looking at cognition and the causes actually that lead the brain not to function in aging and in diseases like Alzheimer's disease, ultimately leading to dementia. I also want to thank Jim Surmeyer because he's really been a great help for this project. And he gave me the opportunity to use the lab and do all the facilities and everything, and his knowledge also of the brain at the end. So just to give you an idea, let me see. Okay, so the basal forebrain cholinergic neurons, it's a group of neurons that, let me close a bunch of things here if I can. Right, so it's a bunch of nuclei that are at the base of the brain. They are distributed rostrocaudally. They are divided in several subgroups. There is the medial septum that projects to the hippocampus and internal cortex, the ventral horizontal limb, the diagonal band of Broca, and the nucleus basalis cellularis. And they are depicted here with the respective projections. So I've been focusing mostly on the nucleus basalis. These neurons actually project to the entire cortex. It's really impressive because these neurons have gigantic axons that go from the base of the brain to the whole cortex. They cover the whole cortex. They modulate, they release acetylcholine. So these are cholinergic neurons. And they modulate the whole sort of things, including LTP, LTD, the neuronal excitability, depending on the receptor obviously expressed in the target cells. And you see here, there is a reconstruction of some neurons. These are 23 neurons that I got from this website where you can reconstitute them. And you see that the projections are huge. They go from here, this is a mouse, obviously, and they go from here to the whole cortex. So, and the idea, our idea is actually that, so it is actually known that these neurons, they get lost in Alzheimer's disease and other related dementia, like dementia Lewy body or Parkinson's disease dementia. These are, they're lost at different times during the development of the disease. They also malfunction, to use an easy word, during aging. So all these diseases, they might have actually a background of age-related impairment of these neurons. So, and the loss of cholinergic markers is actually correlated with the degree of intellectual impairment in Alzheimer's disease. And in enhancing cholinergic signaling with cholinesterase inhibitors is actually one of the few treatments that there are to try to reduce dementia in Alzheimer's disease. And there is no, obviously, there's no cure that can actually fix the disease, but there is some symptomatic treatment you can use. And this evidence actually led to the cholinergic hypothesis of age and Alzheimer's disease cognitive deficits. So the primary risk factor of Alzheimer's disease is aging. And aging is usually associated with major decline of cellular bioenergetics. So the cell is actually less able to cope energetically with the challenges of firing, of activity. And three features that are really important with these neurons that these neurons are spontaneously spiking at rest. So they need elevated energy to keep the spiking, so the action potential and the regenerative activity going, which it's a highly energy hungry process. They have extensive axonal arborization, as I showed you. So they have elevated basal energy demand for these also. They need to maintain these huge axons. The axon is associated with a vast number of acetylcholine release sites, which is another energy hungry activity for that. They need ATP to recycle vesicles in general. So these are three features that render these neurons very vulnerable, I think, and they put pressure on what we thought was what we think is actually the main source of ATP, which is the mitochondria in these neurons. And what we found is actually when in these neurons we use in mito-GFP, which is a probe that you can target to the cholinergic neuron using chat-Cre neurons. So it's a Cre-dependent expressive probe that you can express in these neurons. And this, you see a picture here. I'm injecting the probe directly in the basal forebrain. So I apologize, there's a lot of techniques here. Maybe I don't have time to go through the whole thing, but I hope the message is clear at the end. And you can measure the relative oxidation of the mitochondria in these neurons. So you can measure, you can compare the oxidation of these neuron of the mitochondria in this neuron between the minimal oxidation with DTT, which is a strong reducing agent, and aldrithiol, which is the strong oxidizing agent. And what you get is that these neurons, first of all, they have to begin with a high relative oxidation. So they are highly relative oxidized, like you see in these also younger mice, 7 months and 2 months old mice. And there is no difference between these two. Actually, I found out that at the end, they're quite similar at the end. And interestingly, the oxidation, and not surprising also, it's increased in older mice that in 16-month-old mice. And so this is kind of normal. You think that the process of aging increases oxidative stress in older neurons, I'm guessing. But it's interesting to see that at younger age, with no pathologies, these neurons are strongly oxidized. The mitochondria of these neurons are strongly oxidized. And this could be a problem, actually, with time. And interestingly, this oxidation is reduced by blocking calcium channels, blocking renin receptors, and blocking basal spiking activity of these neurons. So if you kind of block calcium signaling and the spiking activity in these neurons, this oxidation is reduced. I'm not showing the picture here, but I hope it's clear that. And this, we think, is because these neurons, they show the calcium oscillation, they are phase locked to action potential. So I'm measuring these with a 2-photon microscopy, filling the cell with Fura2. And doing that, you can measure actually the calcium oscillation that come together with the action potential, as you see here. This neuron fires usually between 1 and 4 hertz, so they're pretty slow also. And you can measure actually easily, not that easily, but you can measure a calcium oscillation with each action potential. And we think this is actually being found by Jim, actually, other people, that this oscillation signal to the mitochondria. So this, I'll show you a picture after. So you can see that this calcium, when it enters the mitochondria, signal the mitochondria how much energy it needs. So how much is the activity of the neuron in the specific moment. So calcium enters the mitochondria, regulates the oxidative phosphorylation TCA cycle, producing more ATP if the firing rate is actually increased, for instance. The thing is that this process is also producing oxidative species like superoxide here, which increase oxidative stress. So the take-home message of these slides is that we think that these neurons have, like actually the dopaminergic neuron in the substantia nigra compacta, the region that gets destroyed during Parkinson's disease. They have this mechanism that regulates the energy balance of these cells somehow, but it also helps to increase the oxidative stress of the mitochondria across the entire life of the neuron. And this could be something that might be a weakness of these neurons, actually, and it actually gets worse with age. So, and this, I'm switching here, this is mostly physiology of these neurons. I'm switching here to another part of the presentation where I'm talking about the alpha-synuclein pathology. So another hallmark of Alzheimer's disease and related dementia is actually the development of proteinopathies. And we were studying this, an alpha-synuclein model, pathology model, that was showing that, like you see in this figure, I hope it's kind of clear, that when you inject alpha-synuclein preformed fibrils in a specific region of the brain, in this case, there's a PPN, the cholinergic neuron uptake the fibril, and they show a strong pathology inside the cell. As you see here, the white is overlapped with the red all the time. So most of the time, this pathology is only in PPN cholinergic neurons. And the idea of this, this was the basic idea for the grant, was to look at what will do the injection of this preformed fibril in the basal forebrain and see if this fibril gets uptaken by the by the cholinergic neurons, and see to look at the effect of this. And this could mimic the pathological effect that these fibers have in the humans in Parkinson's disease dementia or DLB. So we inject these fibers in the basal forebrain. We wait 10 weeks of incubation, and this is done in younger mice. And what we found was surprising, actually. We found that there is no, differently from the PPN cholinergic neurons, these neurons, they don't develop any inclusion. At least they don't develop any phosphorylated synuclein. This is completely different from what happens to the PPN. And we're trying to figure out what happened, actually, in this case. What's the difference between these two neurons? And also, they don't seem to accumulate also in a PV, positive neuron. There are parvalbumin GABAergic neurons that are present in the basal forebrain. But they are present here. So we're trying to figure out in which neuron these fibrils develop. And we still don't understand. We're not sure what's happening, but it might be glutamatergic neuron. That's our hypothesis. And we're trying to figure that out actually right now. So you see that there are neurons that don't express alpha-synuclein, phosphorylated alpha-synuclein, which is supposed to be aggregated inside these neurons, and it's not. It's another neuron, but we see physiological effect of these fibril injection. So when you inject the fibrils and you measure basal firing rate of the basal BFCN, of the cholinergic neuron basal forebrain, you see that the firing rate of this neuron increases significantly, as shown here. So this is a control. This is the neurons injected with the neuron injected with the fibrils, and this is decrease. You can reverse this effect using two strong antioxidants, N-acetylcysteine and L-name. So indicating that there is an oxidative stress induced by the preformed fibrils that somehow develops in these neurons. And we don't know exactly the mechanism. That's what we're trying to figure out. Another thing that happens is that the oxidative stress of these neurons actually increased. This is the same technique I showed you before. So mito-GFP. So the mitochondrial oxidation is actually increased in these neurons. And again, as I've shown you before, it's decreased with NAC and acetylcysteine. So decreasing the oxidation obviously decreases the oxidation of the mitochondria with an antioxidant. Another interesting thing was that the PFF injections in basal forebrain was able to increase the reliance of these cells on ATP from the mitochondria. So the idea is actually these neurons increased firing rate. The firing rate puts more stress on the energetic balance of the cell, and the cell at this point starts to use the mitochondria more for the production of ATP. And what is interesting here is actually, and I have a slide at the end eventually if you have time, is that if you look at this slide, you see that the way we do it with this measure is that we use a Perceval, so you can monitor the ratiometric levels of ATP and ADP. So you measure how much ATP and ADP is at that time in the cell. And you see that putting oligomycin in the bath applying oligomycin, you see that there is a decrease in ATP, right? Which is increased with the PFF injection. But the level of ATP produced by these cells through the OXPHOS, so oligomycin it blocks the production of ATP from the mitochondria. The level of ATP is produced from the mitochondria is really low here in this case. It's about 20%, which is 20–30%. And if you look at other neurons, like dopaminergic neurons, they have the same, we thought, features of these neurons, like long axons, huge innervation of the striatum in that case. they usually they have this value at about 60–70%. So 60 or 70% of the ATP is produced by the mitochondria. And so this is we thought it was really an interesting feature. I'm also trying to figure this out also on a side project, because this could be interesting from aging Alzheimer's disease point of view, where there is stress, where the metabolism of glucose is actually reduced somehow for different reasons. We don't know exactly why. And this could be that these cells, they can't really cope with the bioenergetic stress too much, and that's why maybe they are vulnerable in Alzheimer's disease. And so one thing that we found with the injection of preformed fibrils was an increase in astrocytes activation. So this is the site that was being injected with PFFs, and you see that there is a high increase of GFAP positive astrocytes compared to control here. You see that the cholinergic neurons in red. And we thought this was also interesting. So this project obviously is opening a lot of more questions that is actually closing, but this kind of interesting. It gives me a lot of loose hands that I can actually try and tie. So there is increased inflammation when you inject these fibers. We think is actually temporary. So we've shown in other regions of the brain that this increased inflammation is temporary, but in 10 weeks after PFF injection, there is still inflammation. And I still have all this effect on the physiology of these neurons, which is interesting. One thing we were thinking was that the astrocytes might somehow speed up the firing rate of these neurons. And this is with the release of mediators like nitric oxide, for instance. So, and I tried this experiment once, and it actually worked, and it's really remarkable in the sense that it's a pretty simple experiment to do, and it gave the result you expect immediately. And so what I did, I injected GFAP with a probe, channel rhodopsin with a GFAP promoter in basal forebrain. So I basically express channel rhodopsin only in astrocytes. And when I stimulate the astrocytes with a LED light at 470 nanometer, I see a strong increase in firing rate of the cholinergic neurons that have been patched with the in cell attach mode here, as you see here. So the firing rate here and then increases suddenly when the LED comes on. And my idea is actually that overall is that the PFF somehow activate the astrocytes. The astrocytes start releasing these the transmitter. I'm not sure what it is, but I'm suspecting is a nitric oxide, which is known actually. I tried, and it's known to increase firing rate of cholinergic neurons, specifically also the basal forebrain cholinergic neurons. And these start changing the oxidative stress or oxidation of the mitochondria, and start changing the way that these neurons produce ATP. And these can worse with age in terms of making these neurons more vulnerable to challenges that happen in Alzheimer's disease or other related dementia. And one thing I wanted to show you was the last slide that I was talking about before. If you look at this, it seems very similar to the other one. In neurons, in a basal forebrain cholinergic neuron, you see that the OXPHOS index, which means the ATP that is coming from the mitochondria, is about 30%, or between 30 and 40%. But in cholinergic neurons, the dorsolateral striatum, which are not lost in Alzheimer's disease or other dementia, the OXPHOS is much higher, similar to the dopaminergic neurons. And this might be that actually the dorsolateral striatum cholinergic neurons might cope much better with the restraint of energy of glucose metabolism that we see in Alzheimer's disease and maybe in aging also. And this is actually insulin dependent. This is actually what I found lately, and this is really interesting for me. So, and this is it. Actually, I forgot to put the thanks slide, but I want to thank you guys, all of you again, and it has been interesting. And I get plenty of data to start whatever actually now. Thank you so much.
Okay, thank you, Trist.
Okay.
Could I ask a question?
Yeah.
So Tristano, very interesting work. I was intrigued by the fact that the PPN neurons and the basal forebrain neurons behave differently.
Right.
Now, in the human, these two cholinergic neurons have different properties. For example, the basal forebrain cholinergic neurons, almost all of them have a lot of calbindin D28K, whereas PPN doesn't. Now, PPN neurons have nitric oxide synthase, which the basal forebrain cholinergic neurons don't. So do you think those differences might explain what you see?
There could. So we did also, which were not too conclusive. We had some issues with the sample and the PPN, but we're redoing this. The pathways is the most affected was actually, so we're looking at differences in gene expression, PPN, and the basal forebrain cholinergic neurons. That could be an explanation, yes. I'm very attached to this metabolism features of these neurons, metabolic features of these neurons, which they see, everything's pointing to the fact, and actually it would be interesting to see your opinion also. Everything is pointing to the fact that these cholinergic neurons in basal forebrain, they are much less relying on mitochondria for the generation of ATP. So that is actually what came out of those studies, which are not completely conclusive yet, but that could be an explanation. Calcium handle of this neuron could be different, and we have no data absolutely on the PPN cholinergic neurons. But one of the pathways came out really strongly was the glycolytic pathway, for instance.
I have a question here. I don't know if you see that, Dr. Disterhoff.
I don't.
Okay. So we do have a question from Dr. Vassar, and he said, I might have missed it, but do the BFCNs degenerate when, I think it's supposed to say they take up PFFs? No, so in theory, yes. So in patients, they found degeneration when they express, but I can get them to express that. So the idea, I thought about the fact that these neurons, when I do the test after the injection of PFF, they just die. I can see them, the inclusions, just because they're not there. But I've done like several mice, and I cannot see any overlap in, let's say, red-green between PFF antibody and the cholinergic neurons. So I'm prone to think they actually don't even pick up the PFFs, or the phosphorylation of the preformed fibrils is actually of the alpha-synuclein, which is what we're detecting with the specific antibody that are shown, is actually not relevant in this neuron. It might be have to look. I might need to look at the conformational of the alpha-synuclein inclusion section with a different antibody. That's another possibility.
Okay, can I ask one, and I guess it'll be a final question, Tristano, and just stepping back a little bit. Most people, when you think of Parkinson's disease, think of dopamine deficits. But so what's the cholinergic contribution in Parkinson's? Is it to the motor change, or is it, but there is also this cognitive change in Parkinson's. Is that what the cholinergic change is contributing to, or what?
Yeah, so a lot of research, especially with GMOs, has been done in striatal cholinergic neurons, and we know that those are involved in dyskinesia. So, and the activity changes with the time during development of Parkinson's disease, so they actually have an effect on movement, and we know that clearly. It's less known what happens in these because so these neurons mostly the basal forebrain cholinergic neurons, they're mostly related to attention and cognition, but they for sure they have an effect on movement also. So and it's not clear what they actually do. But you know, when you have a complex task, you have attention and movement. They work together in order to achieve the goal that you're looking at. So yeah, and they're we're still really not sure what these neurons. It's really hard to study them without any markers that we have now, like tomato-based TD tomato Cre lines and all these kinds of things. They're immersed in a vast number of neurons at the base of the brain that it was really hard to patch, for instance.
Okay. Shall we, Lisa, I think.
Yeah, I think I don't see any other questions unless we have any last minute questions for either Dr. Gratton or Dr. Pancani.
Okay. Well, if not, what I would like to do is thank both Dr. Caterina Gratton and Dr. Tristano Pancani for really interesting, informative talks. And for giving us an update on the projects that the AD Center has funded for you. So thank you very much for your participation.
Thank you very much.
And thanks to the audience for sharing these for listening in. Thanks.
Thank you.
All right, thank you.